Hello everyone, so this is part 2 in the videos on diffusion, and in this one we're going to talk about diffusion in biological systems. Now what I've done is taken 4 classic examples of where diffusion takes place. We've got within the leaf on the top left, the alveoli in the lungs top right, we've got the villi of the small intestine in the bottom left, and a synapse, the gap between 2 nerves at the bottom right. So all I'm going to do is just talk through each one of those in turn and just say how the structure really is maximised for diffusion. The passive movement, random net movement of particles from an area of high concentration to low concentration. Now, in my previous video on diffusion, we talked about Fick's law. And we said that the rate of diffusion was proportional to the surface area times concentration gradient divided by diffusion distance. So in that we're saying a large surface area, large concentration gradient and a small diffusion distance would all increase the rate of diffusion. So we're going to see how those 3 come about essentially in each of our examples. So let's start at the top left with the plant. So in terms of surface area, the leaf is very thin and very flat and wide. So if you have a very flat and wide leaf, then you have a large surface area to allow the diffusion of gases into the leaf essentially and out of the leaf. And when we say gases, we're thinking about carbon dioxide for photosynthesis, oxygen for respiration. So the leaf being flat and wide gives us our large surface area. So that one we can tick, having explained. Concentration gradient, so how to get that nice and high. By having air spaces within the leaf, and I'll just draw a little arrow to show you some of these. So just here we have air spaces between those sort of round cells which are called mesophyll cells. That allows gases to diffuse in and out and within that leaf structure, and having lots of CO2 outside, for example around here, and a bit less within the air space in the leaf allows it to diffuse in. So there's air spaces allow us to maintain that concentration gradient or difference. And finally, diffusion distance. We want it to be nice and thin for things to move from A to B as quick as possible. Now, if I just draw an arrow over the leaf, and we'll do this black just to make it more visible, the distance from top to bottom is really only about 6 to 8 cells thin. It's very, very small. So our diffusion distance essentially from the stomata, which are here, the pores or the holes at the bottom of the leaf, to the top is very, very small, because ideally, remember, we're wanting gases to diffuse through those stomata, those holes at on the underside or the lower epidermis of the leaf, up towards our palisade cells to use in photosynthesis. So by having a thin leaf, essentially with only a few cells thin, or a few cells thick rather, that maintains our short diffusion distance. So the plant is one of our biological systems. Let's move over to the alveoli in the lung. Surface area. Well, in each lung there are roughly 500 million alveoli, which is a very, very large number. And the point of the alveoli is essentially it's the site where we get gas exchange. Oxygen moves from the alveoli into the blood capillaries, and carbon dioxide moves from the blood into the alveoli to be exhaled, essentially. So by having lots of alveoli, we can get, well, lots of gas exchange, really. So that one gives us our large surface area. Concentration gradient. Now, if you imagine oxygen is within the blood capillaries, it's going to be transported essentially to the heart to be pumped around the body to the cells and tissues that need it. And that oxygen diffuses through the alveoli wall into that capillary. Now, because blood isn't stationary, because blood keeps moving at any point along the alveoli, there is always more oxygen within the alveoli in that air sac compared to the capillary. If we just draw a very small sketch just at the top to kind of explain that. If you think of this as a two-layered thing here, now again, this isn't really the best diagram, but I want you to imagine that oxygen is here within this alveoli, and it diffuses through the walls into the blood capillary. Now, if oxygen is simply just moving from the top to the bottom, eventually you'll reach a dynamic equilibrium and then there'll be no further movement. So the fact that oxygen in that blood is transported consistently away from that site, so because blood is being carried through the capillaries continuously around the alveoli and thus carrying the oxygen away, you always have this concentration gradient set up where you always have more oxygen in the alveoli, so in the top, than you do compared to the blood at the bottom. So that's just a little quick sketch to show how we maintain concentration gradient. And finally, diffusion distance. Well, the wall of the alveoli is only one cell thin, and so is the wall of the capillary. So when oxygen, let's say, moves through from the alveoli into the capillary, it doesn't have very far to go. So we have a very small diffusion distance. Now let's think about the villi, so in the bottom left. The villi are finger-like projections that you see lining the small intestine. Now the very fact that you have these villi allows you for an increase in the surface area for the absorption of the products of digestion food into the bloodstream. Now, if you just imagine, and I'll draw this just above, if you take that space there, now you can have food being broken down by enzymes during digestion and being absorbed, let's say, across that sort of gap, that space. Now you can see you're limited by how much you can get through from one side to the other through that small intestine. Whereas if you were to have a space that looks like this, you can see that within the same sort of space across, by having all these foldings, you maximise the surface area. And that's what the villi does. It maximises the space that the products of digestion can get absorbed into the blood into. So that's how we have a large surface area. Concentration gradient, to maintain this, essentially it's like the alveoli in the lungs. By having blood vessels here, and just label them, so by having this network of capillaries free flowing, essentially it means that the products of digestion go through the wall of the villi and then into the blood, and as the blood circulates it carries them away, meaning that there's always more of those products out of the blood rather than inside, allowing the continual diffusion of them into that capillary. So concentration gradient comes from the movement of the blood within that capillary carrying the food products away. And finally, diffusion distance. Well, the cells that make up this villi and of the capillary are only one cell thin. So very small diffusion distance. And finally, we come to the bottom right and our synapse, which is the junction sort of between two nerves. So let's think how in this synapse you would maximise surface area concentration gradient and make the most of the diffusion distance. Well, surface area essentially comes from the fact that you have multiple dendrites branching with multiple axon terminals. Now, a dendrite, as I've mentioned in a previous video on the structure of nerves, dendrites are these extensions that come from a cell body, and then that cell body extended down to form an axon, and coming from that axon you get a load of axon terminals. And an axon terminal synapses with a dendrite. Now, by having that sort of connection or that means of communication, but on every single dendrite and every axon terminal allows for this massive network of neural tissue. And that's how you increase surface area for diffusion, by having this large sort of network of nerves in contact with one another. Now, when we say diffusion, what we're essentially referring to is diffusion of this chemical neurotransmitter, because an approaching nerve impulse, as we've got in the diagram, comes down the axon terminal of 1. These vesicles with neurotransmitter move to the presynaptic membrane, which is here. They bind with that presynaptic membrane, burst, and they release neurotransmitter across the synapse, across the gap. And when they reach a receptor on the postsynaptic membrane, which is here, that triggers an impulse in the next nerve. So by having this kind of setup, this synapse in multiple places allows for a large surface area, essentially. Concentration gradient. Well, if you notice in this picture, vesicles of neurotransmitter are only here. They're only on one side. So you have lots of vesicle with neurotransmitter on one side, none on another. You've got an area of high to low, so diffusion can happen. So we deliberately don't have any vesicles of neurotransmitter in the postsynaptic nerve to maintain this concentration gradient. And that also allows the impulse directionally to only go one way. And finally, diffusion distance. Well, you can kind of see from this picture that the synapse is very, very small. That gap between the two neurons is tiny. So it doesn't take long for the neurotransmitter to diffuse across it. So that's how we obtain our very small diffusion distance. So here are just 4 classic examples of diffusion in biological systems. The key thing really to remember with this is that you're always looking at how fixed law can be applied. How does structure link to function and allow for a maximum surface area, a concentration gradient to be maintained, and to minimise that diffusion distance? Okay, thanks for listening.